Okay, so today I'm going to talk to you a little bit about nutrient and carbon transport in the same streams that Warren was just talking to you about. My research questions going into this were many, but they break down into three basic themes. The first of which is, how do the nutrient and carbon concentrations in these streams compare to concentrations at other locations across the world? What temporal trends are present? And what effects do hydrology and stream morphology, things that OESF is studying, have on carbon and nutrient export at these sites? So as Warren just set up, the study area is in the Olympic Experimental State Forest. It's a large area of temperate rainforest. Obviously, you guys probably know a bit more about this place than I do, actually. Lots of precipitation, and the thing that I think is pretty interesting is this dense network of streams. So there's a lot of water moving things around out here. As far as the soils go, we're in the Sitka spruce belt and the northern Pacific Coast Range. The soils are well drained and deep, which I think, again, is pretty important in thinking about carbon and nitrogen moving through these soils. Moves quickly. These soils are also very rich in organic carbon, and they have high concentrations of nitrogen, but they are variable. And there's a lot of biomass production, which, again, you guys know quite a bit about. So I just want to remind you guys of the hypothesis that OESF is working under. The HCP that was adopted in 1997 is looking to show increased or improved habitat conditions that will reflect the natural variability of these sites. And I think that natural variability is what's really important here, which is why I set up my study to be longer term. And I think streams are really cool because they can incorporate what's going on in the landscape into a single point. So the water falls as precipitation onto the landscape, and as it flows through the watershed, we're able to go to one spot in the stream and get a summary of what's going on at that basin. I also think it's important because the export of organic matter hasn't really been studied very much in this area. Compared to tropical climates, temperate rainforests are particularly understudied, but it's very possible that we have high export of organic matter and nutrients from these sites. And so undisturbed rivers worldwide have ranges of between 16 and 240 micrograms of nitrogen per liter, which is a pretty high range. And that variability has to do with the transformation of nitrogen into other forms besides nitrate. The nitrogen cycle is very complex. And so nitrogen can shift around from different species. And so measuring just one species of nitrogen, so nitrate, gives high variability because the nitrogen can be in a different state. And in general, the highest levels have been found in temperate rivers. Phosphate, on the other hand, has a smaller range in undisturbed rivers. And so what I mean by undisturbed is the quintessential nitrate influencer is agriculture, right? So if you think about fertilizer being applied to the land, it flows into the stream and there's high concentrations of nitrate. Logging can have an influence on nitrate and management at these sites, but it's variable and it's not as immediate as, say, agriculture is. Carbon, so DOC, dissolved organic carbon, and POC, particulate organic carbon, I'll use those abbreviations occasionally here. Between 1 and 20 for the dissolved, and 1 to 25 for the particulate. But particulates depending on how turbid the water is, can have a very high variability. Both DOC and particulate, so dissolved end particulates, are strongly coupled with discharge. And so a higher discharge flushes more carbon into the stream. And they're also very high in temperate and tropical rainforests because of the high rainfall. Particulate carbon, so the more sediment that is in the stream actually reduces the ratio of carbon of organic matter to inorganic matter. So the more sediment you have flushing, the lower you're going to have, the lower ratio will be to particulate organic carbon. So my methods, I went to the 10 sites that Warren just described that are being sampled for or monitored for hydrology within the OESF. I went one time per season, so 4 visits total over the past year. And I measured a suite of geochemical variables, including dissolved particulate and carbon. So the 10 basins are spread out amongst the OESF. This plot here shows 14. And as both Warren and Jeff were saying, monitoring discharge at these sites is very difficult because of the shifting stream channels. And so a few of the sites have been removed from this study due to an inability to accurately measure the discharge over time. And so the locations, again, similar to what Warren was talking about, within the basin are at the near the mouth of the stream. There's roughly 100 meters long, outside of the influence of the main stem. So the main stem isn't adding any carbon back into the stream reach. And the geochemical sample sites that I chose within this 100-meter stream reach were towards the upper end of the stream reach, and part of that is a practical reason in that when I would go to the sites with other technicians, I wanted to make sure that there wasn't a lot of disturbance upstream from people walking, and so I placed my sites further upstream. Just back for one sec. I also chose my sample sites based on the stream morphology. And so I wanted to make sure I wasn't pulling a sample from a pool or a stagnant place or somewhere that had a very obvious influence on the organic matter present in the stream. And so in general, I chose a site that was towards the end of a riffle or swiftly moving water that didn't have any obstructions upstream from the site. And so I went out 4 times. This is just overlaid on top of the Queets River hydrograph, just to kind of give you a reference for what the hydrologic conditions were at the time that I was taking these measurements. And it's actually kind of hard to see here, but there are green lines showing when I went. And so my first event was in June, or the end of June, towards the trail end of the high flow period. In August, we were at base flow conditions, really low. November was the highest flow conditions that I observed. And then in February, we see a return to base flow during the high flow period. So kind of a unique time point. So just a little bit of how we do this. I know that Jeff who's here, kind of told you a little bit about that as well. But for every sample that I collected, we also collected a discharge measurement. And that way, the rating curves that Jeff was showing, we have calibration points that are specific to the geochemical sampling points. And so we can get a good measurement of discharge at that time. This is the schematic that I used in the field to separate all of the different pieces of data that I was collecting, and I'll use it here to kind of explain to you all of the different things that I measured. So I collected stream water and used a sieve to separate out the coarse suspended sediments. I then took that sieved water and filtered it to get the fine suspended sediments. The filtrate from that went on to dissolved nutrient analysis. I then took the sieved water and also filtered it with a different type of filter that doesn't have any carbon in it, a glass fiber filter, to get fine particulate organic carbon and fine particulate nitrogen. The filtrate from that went on to the dissolved organic carbon analysis. I also used a zooplankton net, and these are 0.45 micron filters, so separating out particulate from dissolved. I then used a zooplankton net, which was similar to the sieve in that it was 63 microns, to get coarse particulate organic carbon. And so the combined coarse suspended sediment and coarse particulate organic carbon and nitrogen samples can give us particulate concentrations. So I can get the suspended sediment concentration, then figure out what the C and N content are from this other sample. So here you can kind of see what I have set up. It's hard to take a photo of all these different things. So you can kind of see what I have set up in the stream here. The net was what I placed furthest upstream because I didn't want to have any influence from any of the meters shifting around, kicking up sediment and influencing more towards the benthic sediments. I also used 3 different meters while I was in the field, a pH meter, a YSI meter, which measures conductivity and temperature and dissolved oxygen, and a color dissolved organic matter meter, which we will use to correlate with dissolved organic carbon. Okay, so in the lab, I've been spending a lot of time in the lab. I analyzed these samples myself. And so the nutrient analysis I did on a SEAL discrete auto analyzer. That was a video, but I don't think it's gonna work, and it's okay. Using these different EPA methods to get the nitrate, ammonia, and phosphate concentrations. Suspended sediments, I kind of already explained a little bit of this, but those filters, the pre-weight and the post-weight, is how we figure out how much sediment is in those samples. And I won't cover this, but it's just here. The particulate organic carbon analysis, we're using a Perkin-Elmer elemental analyzer, which goes through a series of combustion and reductions to get carbon and nitrogen content. So I have some preliminary results. I do have a disclaimer, this research is still in progress, so some of the things that I've set you up to want to know about, I don't actually know yet, so you'll have to pay more attention later. So the nutrient values over time, so on the y-axis I have phosphate, ammonia, and nitrate, and the 4 sampling events. And there are some interesting trends over time. This November sampling event, I'll send this to you too. This November sampling event was that high flow, so when the streams were really high, and it shows a very, very high concentration of nitrate in the stream. So remember the range of nitrate in the streams was low, but 200 was the maximum in natural rivers. And here we're seeing a 10-fold increase from natural rivers at these sites. And I'll talk a little bit more about why I think that is later. For ammonia, we kind of see the inverse relationship between nitrate and ammonia, which makes sense. As nitrification is happening, we're going from nitrate to ammonia. So in June, when things are growing, we see more ammonia in the stream. In August, all of my samples were below the detection limit for ammonia, so very low. And in February, we weren't able to measure ammonia. And phosphate has an inverse trend with discharge. And so you can see it's higher and more variable when there's less water around. In November, with those high flows, we're seeing dilution. So rainwater doesn't have a lot of phosphate in it. And so as more water is being added into the stream, we're seeing a lower and lower concentration of phosphate. And so just to show you, discharge over time has very low variability among sites in all of the sampling events aside from November. So as we see those high flows, that's when the variability in those streams really comes into play. So plotting all of the sites against discharge, so this is the nitrate concentration against discharge, doesn't really show a very strong correlation. There is a positive trend. You can see the correlation coefficient is pretty low here. Phosphate, inverse trend, very interesting setup here. As we're seeing more variability in the discharge, that's when all of the phosphate concentrations are low. And when we have variability in the phosphate, that's when all of the discharge concentrations are low. And again, not a very strong correlation. And ammonia, not a very strong trend either. If we separate out discharge by site, however, and look at the nitrate and phosphate concentrations, we do start to see influences from discharge. So this is for site 145, one of the basins. And we do start to see a pretty strong coupling between nitrate and discharge. Phosphate, not so much, but still the relationship gets stronger as you look at individual sites. And so nitrate specifically, so this is again nitrate concentration and discharge, and here's a subset of some of the sites. We are seeing pretty strong correlations everywhere, which is consistent with what we were expecting. Now, the temporal trends that we're seeing, I was expecting to be matching up similar to this paper, Ward et al., where they were looking at nutrient concentrations over time in watersheds draining to the Hood Canal. And so in November, we're seeing mobilization, high values of nitrate. In June and August, February, June, and August, low concentrations, which is also consistent with what we've been seeing. Now, these high levels of stream nitrate, we're not exactly sure why this is happening yet. Like I said, these are some preliminary results, and so you're kind of getting a little bit of insight into how scientific research goes, some of the trials and tribulations. There have been studies that have shown that there is potential nitrate saturation due to harvest in basins, and so there's nothing to take up the nitrate from the soils, and so it flushes into the streams. I really don't think that's what's happening here, though, because there are trees growing. These correlations are much stronger closer to harvest times. What's more likely the case is that these are small streams, so they're tightly coupled with the upland environment, and so the availability flushes straight to the streams, and the storms get them there quickly. Just to give you a little disclaimer, all of these things are still to come, so at my next talk you'll be able to learn more about all of the particulates and the suspended sediments and the carbon concentrations.